So I'm Guy McKhann. As I said, I'm a neurosurgeon at Columbia. One of the things I get to do is I specialize in awake brain surgery. So I get to operate on patients while we're looking at their brain, while they're awake, while they're functioning. And the more you do that, the more you realize how little we understand and actually know about the brain. So without further ado, we're really privileged today to have a tremendous group of participants. First, we've got our first guest is Professor of Biomedical Engineering and Radiology at Columbia University, a member of the Zuckerman Mind-Brain Behavior Institute and Kavli Institute for Brain Science. Please welcome Elizabeth Hillman. Our next participant is an assistant professor of psychiatry, neuroscience, and physiology at the NYU Langone Medical Center, who investigates the neural circuitry of mating and aggression in mice. Please welcome Dayu Lin. Also joining us is a neuroscientist and psychiatrist at Weill Cornell Medical College. As a psychiatrist specializing in the treatment of complex mood disorders, please say hello to Conor Liston. And our final participant is a neuroscientist, director of neuroscience at Cold Spring Harbor Laboratory, who is interested in how brain circuits give rise to behavior. Please welcome Tony Zador. Maybe first we could talk about a little bit the history of where did optogenetics come about from, and then you guys can talk about how you're using it a little bit.
So interestingly, actually the history of optogenetics can be traced back to an idea that Francis Crick, who's famous for the double helical structure of DNA, first came up with. He proposed that it would be really convenient if we could activate neurons with light. He proposed that something like 30 years ago. And people had tried different approaches and were developing different strategies for trying to make it work. And it was really the fortuitous recognition that a protein from algae could be expressed in neurons and endow them with the ability to respond immediately to light. And that happened, I guess, about a little over 10 years ago. And it transformed how we do neuroscience. When I started in neuroscience, everything in terms of trying to understand behavior was correlational. We saw neurons fire, and we saw the behavior, and we put them together. And we of course all recognize that correlation is not causality, but without the ability to sort of dissect out the circuits that were responsible for the behavior, all we could do is keep trying to find those associations. And so that happened about, I guess, 2005, and within a couple of years, lots of labs. It happened with it was a paper from Carl Deiseroth and Ed Boyden, and within a couple of years, dozens and soon hundreds of labs were using it. So that's really the basic history.
Great. So Conor, you spent time in the Deiseroth lab before moving to Cornell. So tell us about how you've gotten with optogenetics to what you're doing with it and some of the potential applications as you see it within your world.
Yeah. As you guys heard, I'm a psychiatrist clinically. And one of the things that really excites me about optogenetics is this potential opportunity to understand the neurobiological basis for mental health and mental illness and hopefully develop new treatments. And I think a big limitation in psychiatry is that the drugs we use, I always like to emphasize that the drugs we use, they do work, and they've been a great benefit for a lot of people, but they're blunt tools. They bathe the brain, and they change some aspects of brain function in a way that helps, but they probably have lots of other unintended side effects. And optogenetics affords us this really unique opportunity to really precisely manipulate specific brain circuits, figure out what's going wrong, and try to fix it. And that's one thing that really excites me.
So give us an example of how you've done that.
Yeah.
A specific circuit that you've targeted and what you've been able to find from that.
Yeah, so during my time at Stanford, this is like a collaborative work with several other investigators there. We were really interested in understanding how the brain supports reward-seeking behavior. That's a thing that's obviously altered in the brains of many depressed patients. And we were able to precisely manipulate the activity patterns of specific cells in a region of the brain called the prefrontal cortex, and begin to understand how it regulates the processing of reward-related cues and motivates in animals, and presumably something similar is happening in people's brains, motivates their desire to work to obtain rewards. And interestingly, what we found looks a lot like what goes wrong in the brains of depressed people. There's this abnormal pattern of increased connectivity in this brain circuit, and that seems to be driving some of the reward processing deficits that these rats have, and possibly also people.
So, Dayu, you're using a lot of optogenetics. So what are you using it for, and what types of questions are you able to ask with optogenetics?
Sure, yeah. So I was lucky enough actually to be one of the first person who benefited from the optogenetics. I remember it was still in the days when I was a postdoc, and I have to say, like, optogenetics, although it's a new technique, but the concept-wise is not new, in the sense that neuroscientists have tried very hard for centuries trying to manipulate the brains in order to see what happened. So, but the conventional manipulation is by passing some currents into the brain, so the electric stimulation. And that's where I started when I tried to study the brain before the optogenetic era. Electrical stimulation, there is one deficit of that, which is that neurons, they're not just a cell, which is a ball, but they actually have a lot of processes coming out. So when we electrically stimulate it, we not only stimulated this cell, body, this ball, but also all the processes coming from it. So potentially the cells which is far away can also be activated because of this electric stimulation. And the optogenetics is advantageous because it allows you to really target the cells you're interested in without affecting those cells, just sending processes to it. That's this specificity that makes this tool really unique. And now we're putting the optogenetics into those cells and stimulate.
So why don't we look at if we have the aggression video?
So this is a mouse. There was this part of the brain. It's very deep, very ventral, and we turn down this area, and you see that he start to attack this glove. And he really, that's not what mouse typically do. So they don't usually attack gloves. But in this particular case, when the light activated, he repeatedly sink his teeth into this glove. In fact, when we take it out, this glove is in fact deflated. So, and it's not only gloves. In fact, they will attack other conspecifics, including a female mouse that a male will typically not attack at all. So this inanimate object probably shows the extreme of these behaviors, that this control of these areas just turn on this attacking instinct immediately.
So there's some specificity to the aggression.
It does. It does. In fact, one of the big questions we are trying to ask in the lab is, when we do activate this area, is this just the abiding events, or it's actually increasing this aggressive motivation of the animals? And that's why we're designing different behavior tasks in order to dissociate this part. But that's exactly trying to ask, is it to just make them bite, or actually they has this urge to aggress?
Can you get them to attack a cat?
I would try that. I know how to attack a cat will react to that.
That's a much more complicated question. So all of you guys are talking about a technique where you're able to select out a very small, specific part of the brain to try to isolate and look for behaviors. Now, Elizabeth, you've been focusing on the one hand on using imaging techniques to look at much broader patterns of activity and larger connectivity patterns between the brain and even whole brain activity in various types of animals. Can you fill us in on some of the things you've been doing along those lines?
Well, so we do a lot of technique development in the lab, and I was trained as a physicist, so I did a lot of optics, and so, these technologies, when they come along, they don't just come along. Wonderful people conceive of them and piece them together, but then using them and figuring out how to do that is really challenging as well, and that's a really important factor here. So the little mouse had a thing on its head, and that was actually holding an optical fiber that was allowing you to put laser light into the actual brain to turn these cells on and off. And so in our work, we're really interested in how can you do that better. I mean, it's not just that you can turn cells on, but you can actually turn cells off as well. And the dream is to have many, many different colors so that you can be very selectively turning one thing on and turning one thing off. And that really lets you, figure out what part each one of those things is playing. I was thinking of the analogy of sort of what is my role in getting my kids out of the house in the morning, right? Well, how do you tell that? Do you just throw me in there and make me flash? No, but if you take me away and then see what happens when I'm not there, it's quite helpful way of figuring out what it is exactly that I contribute. And so, that's it's not that much actually, so figuring all of this out. I mean, for me, we do our own experiments as well, and I will talk about that more. But figuring out, working with you guys, what exactly experiment do you want to do? What behavior do you want to do now? Which areas of the brain do we have to get to, and how do we have to make that all work together so that we don't have to have 10 lasers and all of this stuff happening at the same time? Is one of the things that really gets me going. And then we combine that with imaging, because what you've seen mostly there is the readout. You change something in the brain and you look at the way that the person's actions change or the animal's actions change. But we can also simultaneously be looking at what effect that has then on the firing of the other neurons. And so we do a lot of work trying to capture activity at the level of a few cells, at the level of the whole brain. We look at that activity in flies and worms and little fish, and then we also do it up at the scale of mice, and then sort of project that up to the scale of the human brain also.
So maybe it would be a good time to look at the video that you have in terms of patterns of activity, because it's very different from exciting a few neurons with one fiber. You're looking at what the brain is doing spontaneously in its own pattern of activity.
So what you're seeing here is actually the entire top surface of a mouse's brain, and that reveals a whole ton of different areas of the brain that we can look at all at the same time. We're using something called GCAMP, which is a calcium-sensitive fluorescent protein. So what we were just talking about was optogenetics, where you express based on the genetics of the cell, you make the cell produce this light-sensitive protein. And one of the other major tools that has really revolutionized neuroscience in the last 5 to 10 years has been these fluorescent proteins, like the glowing jellyfish that you've probably seen, or the newspapers with the glowing mice, right? So these are calcium-sensitive fluorophores, which means that actually when the neurons fire, they actually flash, and we can read that out. Okay, so it's sort of the opposite. So we've been observing this in the mice, and we've found these amazing patterns of spontaneous activity that tick around in the brain. And what I thought would be fun to show you today is that we do a lot of quantitative analysis on this, trying to understand the cadence of these patterns and the locations of these patterns and what do they mean. And one of the things that we just were able to do, if you go to the next slide, so we found that again, it's hard to see with these patterns. So on the top left is just a still of these kind of weird patterns of activity that we really see coursing through the whole brain. And we found that they were sort of coming on and off in different regions of the brain. And so I'm going to play a movie here where what we've done is, rather than just showing you the movie of these patterns that seem to kind of roll around in the brain, because there's different regions that are lighting up one after the other, we've actually encoded them in color and also as notes on a piano. So what you're going to hear is kind of what it sounds like in the brain of a mouse, as a mouse is just sitting there resting. And this is over pretty much the entire surface of the brain. So all the regions responsible for seeing and hearing and motor actions. This was all done by my student Nick here, who is a biomedical engineer / composer. So if you want to just click the, I'm not kidding, there you go. So every time a region lights up, you hear a note. Give it a sec. So we use this method just as a way to try to just really capture.
Is the relationship between the brain activity and the music fully automated, or is there?
Yes, this is actually using the NMF non-negative matrix factorization algorithm on this data, and then taking the factors and then putting them into musical notes. And then if you go to the next slide, then
just before you go there, I would argue relative to what Tony was saying before, that at rest, at least based on the music pattern, the mice are quite calm.
Well, after I started doing this analysis, I remember I had a big grant on this that I submitted right before Thanksgiving, and I was sitting there in Thanksgiving, and I just kept feeling this rhythm of my brain, and thinking, if I could just sort of calm down and watch some television, if I just watch, 10 Things I Hate About You one more time, like maybe I could get myself into this state, you know what I mean? And so this drew us onto the next part, which is we then gave the animal caffeine. Okay, so you go to the next slide and play the movie on the left. So it's not so different. It's a little faster, has some gaps. Yeah, it feels a little different.
Not enough caffeine.
Okay, and then so we gave it ketamine. So ketamine is a drug. Some people use it recreationally, and they shouldn't. And it's an anesthetic, which really it makes you basically unconscious, makes you high, has various effects of making you forget, and so on. So that's the one on the right here, and you'll see it has a really profound effect. And what's really interesting about this is that this is a very odd modality that we came up with. We actually just take this data using LEDs and cameras. It's very low key compared to the really high tech stuff that we also do in the lab. But the reason why I'm really excited about this is I think neuroscience for a long time has zoomed really far in on just the cells, looking at single cells and how do cells interact with each other. And when we discover this activity that's really everywhere, it really changes the way that I'm now trying to think about, the brain is just sort of talking to itself. It's like the internet, right? It's not just you and you sitting there. It's you connected to all your Facebook friends and all of those things kind of interacting together. And so we're just we're having fun with this, of course, but we're really using this now to try to understand how the whole brain contributes to behavior, and particularly here we're thinking about state. So a state of being anxious, a state of being calm, and really what does that look like, and then how would each one of these react differently if you presented it with an aggressive glove,
You're showing these amazing patterns of how the brain connects and what connects to what. Tony, you've taken a totally different approach to try to study major areas of how what actually connects to what and how we unravel that. Maybe can you explain that for us?
Yeah, so sort of to put it in perspective, when the optogenetics revolution hit, my lab jumped in whole hog. We were already training mice, or at that time rats, to perform what we thought were cognitive tasks, and we could talk more about that. But we were training them to listen to sounds and make decisions based on the sounds they heard to get rewards. And we were trying to understand the circuits that were involved in making those decisions. And we had identified certain subsets of neurons using optogenetics later. We'd gone beyond simply recording the activity of these neurons and actually shown that one set of neurons, it turns out, that project to the auditory striatum, were actually really important in carrying information about a sound toward driving movement. But what I realized from that was that we can make guesses about the circuits involved, and using optogenetics, we can actually test whether our guesses are right. For the first time, like we could test at the circuit level which subset of neurons within a particular area are driving this behavior versus that behavior and what roles they play. But the problem was that we just had to make educated guesses. And one of my students was really fantastic and made a really educated guess and guessed right and showed this particular pathway. But other students made less successful guesses. And the cost of guessing wrong is a couple years of work. You do a set of experiments, and if you guessed wrong, the answer was nope. that wasn't the right guess, and you learn very little.
Time for a new career.
Not my career. Their career.
But for the students,
Yeah,
right?
I used to tell people, I still tell them who enter my lab, I quote from Shrek always. There's a for those of you, my son when he was younger watched Shrek over and over again, and there's a scene where Lord Farquhar sends his knights out to find the princess, and he says, "This mission is dangerous. Many of you may die, but that's a risk I'm prepared to take." if we guess wrong, they're the ones who come up empty-handed. So anyway,
So how did you make it better for them?
So the idea was that we needed a way to screen through the guesses. We needed to know what the circuit was in order to know what was a reasonable guess. Knowing the circuit would not tell us how it actually works, but it would rule out all sorts of possibilities. And the problem was that the tools that were available were either not high enough resolution, didn't have single neuron resolution, or they were incredibly slow. So what we needed to know was whether there are neurons, let's say in my case in the auditory cortex, that send their axons to this region and that region, and whether there are other neurons that send their neurons here and there. And the problem was there was no way of knowing in sort of a high throughput way what the millions and billions of neurons, well, millions in a mouse, billions in a human, where they send their axons. And so the traditional methods for asking those questions were all based on microscopy. The typical high resolution method is to take one neuron in one animal, fill it with a fluorescent tracer or some other kind of tracer, and then track that trace all throughout the brain. The problem is, one neuron per mouse, lots of neurons, that's very slow. If you try to do more than one neuron, the problem is that the processes start overlapping, and you can't resolve where the individual processes from these neurons go. And so the idea that I had 8 years ago now, but it's finally working, was to replace the usual way of visualizing the neurons, which is just to look at a fluorescent dye, with what has turned out to be a fantastic marker, which is DNA, or in fact RNA. So what we do is we cause each neuron to express a unique random sequence of RNA, which then gets transported all throughout the cell.
But how do you do that? Why does a neuron not
So
want to take
real
up
neuron,
two RNAs?
Yeah, in real life, most neurons do not express random. In general, you don't have a lot of random labels floating around your body. In fact, the only case in our body that we know of where we have random labels is your immune system, where the genetic material gets scrambled each time to make antibodies. So we thought about actually trying, and we're still kind of working on trying to trick neurons into scrambling their DNA, but an easier way to do that is we can make viruses that are that each express a unique random 30 nucleotide, 30 letter string of DNA, and then we just squirt that into the brain, and each neuron takes up one viral particle. Sometimes they take up two, and we could talk about technically whether it's not really a problem. And then it gets amplified within the neuron and gets transported out to the axon. So now we no longer have to carefully trace each little process. We can just use DNA sequencing technology, which has transformed other fields of biology. We can just piggyback off of these tremendous advances in DNA sequencing technology, which have driven the price of sequencing our genome down to below $1,000. We can use literally that same technology to figure out where all these neurons project.
So every neuron has its own individual
Every
marker.
neuron has its own individual label. And it's actually, I should have shown this picture. It's inspired actually by an idea that scientists at Harvard, Josh Sains and Jeff Lichtman, came up with about 10 years ago, called Brainbow, where they cause each neuron to express not one color, but a rainbow of colors. And so basically the idea is to replace a rainbow of colors. It's hard to read out more than a couple colors, so there wasn't such a big win, but it makes beautiful pictures. To replace those with these sequences. And so now what we do is we inject into one side in the brain, or in fact we can now tile the entire brain with this virus and figure out where all the neurons in the cortex project to. So in fact, I was looking at your pictures, and we're just now starting to analyze this whole brain connectivity map, and it turns out that there are sort of communities of neurons that talk preferentially to each other. And in fact, the communities that we find based on this connectivity actually are very reminiscent of the sort of different chords, especially in the ketamine brain,
Yep.
where it looks like there's sort of within a community, there's a lot of conversation, then it passes the message to the next community.
So it's possible that you may be seeing with nerve axonal labeling some of the same
I
patterns.
think actually the substrate for what you're seeing is precisely.
We should hang out.
Yeah, we should.
So how do you take that to the next or a different level and say, now we know how things all communicate? How do you start studying what's normal and what's abnormal?
Right. So the idea here is that we can now, for a couple $1,000, figure out the whole connectivity of a brain. There are some issues about resolution. It's single neuron resolution, but the spatial resolution is limited. We have some ideas, and actually we're working on methods that give you higher resolution. But the real interesting idea is that we can now compare the wiring diagram of a normal mouse to a mouse that has a genetic deficit that has been associated with a human disease. And there are now dozens or hundreds of these mice, these so-called animal models of autism and schizophrenia and depression. So we know that there are genes that are associated with human conditions. We know that when you disrupt those genes in mice, they disrupt behavior in ways that are sometimes similar, sometimes not. But in many cases, we think that what's going on is that disruption of those genes causes some kind of change in the neural circuit. But once again, the traditional approach to figuring out what that change is to take an educated guess, burn up a couple of grad students, and if you guessed right, but that is how these things go. It's incredibly frustrating. And so the idea is now we can actually take one of these mice and say, look, here is the disruption. We're not sure which of these disruptions are causal in the behavior, but this totally sort of constrains the set of hypotheses that now physiologists like me and the other people on this stage have to consider.
So you're taking a lot of the guesswork for the grad students out of picking a project.
That's right. That's really my goal, to sacrifice fewer grad students on the altar of bad guesses.
Which is a very admirable goal. We would all agree. Conor, you've taken a different approach to try to look at populations of neurons by putting little prisms in the brain and combining it with optogenetics. Can you talk about that and what it allows you to do that's different from traditional optogenetics?
Yeah, so this work is really similar to what Elizabeth was just describing, but it's kind of on a more microscopic scale. So we're using the same kind of calcium sensors that fluoresce in proportion to their activity, like you saw in those beautiful videos that Elizabeth showed us a few minutes ago. But we are looking with single cell precision on a much smaller area of the brain. One of the functions that we're really interested in is working memory. This basic kind of fundamental form of short-term memory that we all use all the time. Like if we decided we wanted to take a break now and order some pizza, working memory is like what would enable us to kind of look up our favorite pizza place on your phone and store the memory of the phone number long enough to dial it, but it goes away rapidly. And
it's already gone away with the other cell phones.
I give you 3 numbers and you 4.
Yeah, exactly. Teamwork. And it's disrupted in depression and in schizophrenia and in many other psychiatric conditions in different and interesting ways. So we think it's kind of a fundamental process. We could show that that video. So what you're seeing here are cells in the prefrontal cortex. Those black kind of bands are blood vessels, but these are individual neurons, brain cells that are expressing this fluorescent calcium sensor. And that starry night effect is basically a readout of how active each one of these cells is over time. And what you don't see is that this mouse is awake and it's behaving. It's performing a working memory task while we're getting this readout. And so what we hope to be able to do is test hypotheses about how particular cell types in this image that you're seeing here, how they interact to encode and maintain a trace of the working memory during a delay period, during the time it would take us to dial the phone number in that analogy, and how these different cell types interact to kind of imbue working memory with interesting properties. And then once we understand that, like we were just discussing a minute ago, we can formulate hypotheses, guesses, and actually test them using optogenetics to manipulate the activity of specific cell types and test whether our predictions are actually true or not.
So everybody here is using different animal models to study these things to try to come up with information that can help us fundamentally understand human behavior. So maybe as a group, whoever wants to can talk a little bit about how do we best do that? How do we best do that in a way that we are, minimizing use of animals, being compassionate in that, but yet understanding what we need to understand. So what are your guys' thoughts on that?
We recently started using fruit flies, and I don't feel bad about that at all.
So tell us, what can you learn out of a fruit fly?
The beauty of the fruit fly model is that you can breed them really, really quick. And so these genetic techniques now that are really the core of neuroscience that let us label these cells different colors and change them, introduce genetic diseases, make them express optogenetic channel rhodopsins. You can do that in the space of a week. You can just cross two flies and then you have another fly and it's got rainbows in its brain and it does all of this stuff. For me, what's really nice, because when I build microscopes, what you saw there was about a 10 millimeter field of view, which is what we needed, but we couldn't see single cells. But if you have a fly's brain, we can fit that whole thing within our microscope's field of view, and so we can see every single cell in the entire brain. And so we do these completely crazy things where we get the fly and we actually have it walking on a ball and we're imaging its whole brain while it's on a ball, and then you can actually puff odors and odors of lady flies, and then you can see what it does, and you can see where it crawls, and then you're seeing all of this activity in the brain, as it's doing that. And so are flies the same as humans? Probably not. I mean, they can fly, which we can't. So I don't know. But what we're trying to do on so many different levels is understand building blocks, understand basic principles, understand, how does behavior emerge from all of these little cells flashing on and off. And so doing it in that system is a very efficient, very good, nice way of understanding something at that level and then scaling it up. So there's people in neuroscience working at every single level. What we really need to do is all talk to each other and sort of piece together all of those clues. And we also do the maggots as well. I mean, it's gross.
Dayu, you showed us that you work on aggression, but you also work on maternal instinct. So another thing that's across the animal kingdom, but that we all think of as an incredibly human thing, but obviously it's much across the animal kingdom. So maybe you could talk to us about how you study that sort of thing with something like optogenetics.
Yeah, they are, in my views, a lot of social behaviors, instinctive behaviors, they're all controlled by those Asian structures. The mechanisms is very conserved. And the maternal and the paternal behaviors, in fact, parental behaviors in general, in fact, is also highly sexual dimorphism. And one of the regions we study is controlling the maternal behaviors. So I'd like to show you a video here, just to say that we're not only looking at the behaviors, which is a fighting, but also this just heartwarming videos when you look at it. So this mouse, this is a virgin female that she doesn't really care about the pups so much, and she doesn't have much experience with the pups. So this is called a sham stimulation, basically no stimulation. We're just in these areas, then we put a pup in the corner, and she basically ignored the pups. So the pups is wiggling around, probably making a little bit of sound, trying to get her attention. So this part will be 60 seconds, which I guess we're just trying to convince you that there's really nothing happened. So now pay attention. So now we start to stimulate this part of the brain right here. And then she walked right out and bring the pup back home. So they are part of the brain regions which are really just controlling those maternal instincts. And when it turns out, she become instant mom. So
super mom
and do this all day long. Yeah.
So before we move, because a lot of you guys are doing not just mouse modeling but also doing human work too, can you all comment on, because I don't know if anyone here is actually working with it, in addition to optogenetics, out of the Diceroth lab, another great development has been this technique clarity, the ability to take the brain, which is an oblique structure, and essentially turn it into a window pane. So can you all talk about how you see that as a tool, the strength of it, and can you potentially merge it with some of these other techniques?
Well, I think that, so for those of you who haven't heard, there's a technique that allows, so we'd like to see where all the neurons are. We'd like to see how they're all connected. And what limits it is the fact that the neurons are embedded in all sorts of fat and extracellular stuff. And when you do microscopy, it's very difficult to see clearly what's going on. And so for several decades, people have been trying various methods for clearing tissue, for making it transparent. And there have been a couple of major advances in the last couple of years, including the clarity technique from Diceroth's lab and some other techniques that have made it much easier to see a lot of the brain clearly, to be able to image down 1, 2, 3 millimeters. In fact, as the limits now have less to do with the actual tissue and more to do with microscopy. I think that really is making it a lot easier to quickly see what the circuitry is, how different parts of the brain are connected. We're using sort of related techniques. In fact, we're using a technique developed by a guy named Ed Boyden at MIT, which in some sense goes one step further. It allows you to not only clear the tissue, but simultaneously to expand it. So some of the structures we're interested in, it's called expansion microscopy, surprisingly enough. So some of the structures we're interested in are below the resolution of light. And the usual approach to resolving a structure that's below the resolution of visible light is to use electron microscopy, which is a lot of work.
A lot of grad students.
It's a $1 million machine, and just it's a nightmare. At least on this scale, for applying electron microscopy to sort of macroscopic structures. And so what he had, I mean, Ed is actually the developer of channel rhodopsin along with Carl Diceroth. What Ed realized was that instead of making microscopy better, so you could see smaller things, it would be just as good as if you could make the smaller things bigger. And so he developed a technique that was inspired by and actually uses literally the stuff that they make diapers out of. And you know how for those of you who are familiar with diapers, you add liquid, we'll say water, to a diaper and it expands. And so he embeds this diaper stuff in between the bits of neuron, and then binds every little protein to a bit of it, and then expands every protein isotropically in all directions. And so you can get expansion factors that are 2 and 4, and now even 20 times the original. So you can take a synapse, which is below the resolution of light, below 0.2 microns, and make it a 3 micron, 5 micron structure, which we can easily resolve. And I think that technique is really on the verge of transforming a lot of what we can do.
And can you merge that with
So
your
we are
technique?
actively working with Ed's lab to merge our approach. So all of this, just to be clear, is in fixed tissue and tissue from the brain after it's out of the skull. So the optogenetics is fantastic for working with a live animal, but then to figure out the circuitry of that animal, that what you have to do is take it out and then use one of these methods. So we are barcoding neurons while the animal's getting ready to perform a task, and then we expand the tissue and do the sequencing in situ, so we can actually sequence the barcodes while the barcodes are still inside the brain, and it gives us the spatial resolution to actually see which barcodes are at a synapse and actually figure out which neurons are connected to which other neurons. So there are some engineering issues, throughput issues, but conceptually we can expand an entire brain and figure out the wiring diagram of an entire brain at synaptic resolution. And while we're at it, we can also figure out which genes are being expressed. So this is, one of the people in Ed's lab calls it the Rosetta Project, because basically we can figure out everything about a brain, all the way from activity while the animal is performing a task, the behavior, and then the circuitry and gene expression that cause those things. So that's where we're hoping to go with them.
That's pretty amazing to think about. So, but as you say, that's finding out everything about a mouse brain, right?
Yeah.
And so we're talking a lot about, do mice have human behaviors and things? Elizabeth, can you talk a little bit about, you that have been a very strong proponent of that? We've already got great human imaging techniques that you're using in rodents, but also in humans, and trying to study the differences and similarities and what we can gain more out of our MRI techniques we already have, and how you're going about that and helping translate ideas.
Sure. So, I touched on this a little bit before, but has anybody here had an MRI scan? Yeah. Have you ever volunteered for a functional MRI scan where they showed that you had to do tasks and things like this? Okay, so I've done lots of them. I fall asleep. I'm a terrible subject. But you go into an MRI scanner. It makes this beautiful image of your brain. And what they discovered about 30 years ago now is if you actually took a sequence of images over time, that you could see small changes in the signal in different regions of the brain while you were doing things. So like if you actually moved your hand, you could find a region of the brain that would have some sort of correlated change in the MRI signal while you were doing that task. And we call that the BOLD signal, the blood oxygen level dependent signal. And it's what generates those beautiful images that you see on the front of the New York Times saying, this is the area of the human brain that reacts when you see a picture of a puppy or something. And so, I did my postdoc up in Boston in one of the centers that actually developed this method. And so right now, this is the best method that we have for looking at the human brain. It's non-invasive. Any one of you can go sit and do one of these studies and pretend to be a mouse, tapping your fingers or being shown pictures, and it does a pretty good job. It points to this area here. Something happened here, right? But the problem is that signal actually comes about because there was a change in blood flow in that area. So there's not really a change in the MRI signal when a neuron fires, but there's a change in the magnetic properties of blood when it becomes oxygenated or deoxygenated. And so that little signal change that you see is actually coming from a change in blood flow to the area. And that's why it gets a little bit confusing, because it's a little slower than the neural activity because it takes a while for blood flow to change after neurons fire. And so I've spent, in addition to the other work I've done, I spent a long time studying the relationship between neural activity in the brain and the blood flow activity that kind of accompanies it. Partly initially to help with understanding those fMRI images better, functional MRI, because we knew you're seeing blood, but we wanted to understand what that meant in terms of neurons. I could talk about this all day, but it caused us to have to develop a lot of imaging methods to go in and use microscopes to simultaneously look at neurons firing and blood vessels next to them. And I got very interested in it from the standpoint of it's actually quite surprising that every time a region of your brain has some neural activity, that there's actually a big increase in blood flow in that area. And that has to happen for a reason, and that has to be an important part of brain health. And so now my work has sort of moved towards not just trying to understand the fMRI signal better, but also recognizing that there might be situations in which that relationship between neural activity and blood flow gets disrupted. And if every time you're hungry, you try to eat a sandwich, but you miss, something's going to happen. You're going to lose weight. You're not going to perform as well. And so all that stuff I showed you with the coffee and the ketamine, really the core of that study in the beginning was looking at how that neural activity actually was coupled to blood flow changes. And we've got ways now we actually can give drugs that disrupt the relationship between neural activity and blood flow, and we can then see what effects do they actually have in real time on neural activity, what effects they have on behavior. So we're training the animals to sit and actually push a lever. This is one of Nick's other projects, right? Flick their whisker, try and get them to push a lever, disrupt this blood flow relationship, and see whether they actually become slower at pushing the lever. simply because this is just another aspect of the brain that we really don't understand. Stepping back, if we can get a better handle on this relationship between blood flow and neural activity, it gives us a way to really better use functional MRI. And this method has kind of an image problem, right? We all agree. Because it's not so clear what it means. And in fact, when you do it in someone who just had a stroke, you might see huge activity in an area where they're paralyzed, or you might see no activity at all and they go on to have a great recovery. And so sort of my mission is to use these methods more in the mice, where we can do this very detailed imaging, but to try to disambiguate all of this. Try to find, is there an early signature of Alzheimer's? Is there something that happens in stroke that we can actually understand in terms of what it's going to look like in fMRI? And then can we use fMRI in a much more guided way to get to those conditions in human brain? So it's sounds a little circuitous, but it keeps me busy.
I think we all agree it's something there's a huge need for, to understand what is it we're seeing and how do we learn from it. Conor, you've done some of the same thing in trying to tease apart subtypes of depression using functional imaging. Can you tell us kind of how you've approached that and what you've found and what you think the potential for that kind of analysis is?
Yeah. We're really excited about this. Is another area we're really excited about in my lab. The opportunity to potentially use brain imaging to rethink the way we diagnose psychiatry. If you'll make diagnoses in psychiatry, if you'll bear with me for like 2 minutes, I'll tell you an interesting historical little background story. So our DSM, this book that we use for diagnosis, is kind of seeped into the public lexicon, right? You read about it in the Times. A lot of people are familiar with it. And the whole way we diagnose mental illness is really not very old. It was completely changed in 1980. I was born in 1980. You can't say that about very many other areas of medicine. And yet it's the truth. Everything we do in psychiatry is quite different from before 1980, due in part to this kind of controversial study that was published in Science 7 years before that. It just showed essentially that our diagnostic system at the time was not very reproducible and very prone to bias. So a social psychologist at Stanford very cleverly designed a set of experiments where he sent his investigators into hospitals around the country presenting with a fake symptom. They said, I'm experiencing voices saying the words empty, hollow, or thud. And otherwise, they were supposed to answer all questions honestly, truthfully. And the results, they just followed them. And the results were really shocking to everybody. They got different diagnoses in different places. Later work showed that your socioeconomic background and your ethnic background could bias the way people assigned diagnoses to you. And it was really hard to detect that these were fake patients, basically. That's what he showed. You could bias people's judgment in that way. And then in a second part of the study, and this is kind of the clincher, in a second part of the study, the lead on this study had met the director of a famous hospital at the time, and he said, this is an anomaly. What you found is not representative of the way we do business. Come to my hospital, see if you'll get different results. And so they agreed he would send an unknown number of people into his hospital over the next few months. And during that time, their job was to figure out who is a fake patient, who is a real patient. 20% of people were identified as definite imposters. Another 20% were identified as probable imposters. So fully 2 out of every 5 people who came to the hospital were thought to be possible imposters. And the truth was that he hadn't sent any imposters in. They were all real patients. So you could bias people's judgments in both ways. And so getting to the point here, the DSM-3, which was released in 1980 and very similar to the one we use today, was really designed to lump lots of people together with very different problems in big diagnostic categories that we could all agree on. And it did a really good job of that. The diagnoses are much more reproducible than they used to be, less prone to bias. But the problem is they don't correspond to biology very well. So depression is a great example of that. You have this choose 5 or more of 9 symptoms. So there's at least 256 ways you can be depressed, and some of them are almost opposites of one another, like sleeping 19 hours a day, can't get out of bed, not enjoying any activities, very slowed, gained a lot of weight. That person's depressed. So is a person who is sleeping 3 hours a day, anxious, weight loss, almost the opposite. And they get the same diagnoses, but they probably don't have the same biological problem. And it's a miracle, really, that our treatments work as well as they do in these very different people. So what we're trying to do with fMRI is figure out whether we can identify clusters of patients that have similar biological problems as indexed by fMRI, and which can be used to map functional connections in the brain. And if so, then maybe we can target specific treatments to individual people who are most likely to benefit from them.
And can you find different patterns of patients based on fMRI?
Yeah, so we found. I always emphasize I don't think this is the best or final solution to this problem, but it's one solution that works well. We found we can identify 4 very different patterns of abnormal functional connectivity in the brains of different depressed people. We can assign individual people to those categories based just on their brain scan, and then those categories predict different kinds of symptoms and a different likelihood of responding to transcranial magnetic stimulation, which is a brain stimulation based treatment that you could imagine it. I mean, it's on a much coarser scale, but the goal is the same as with optogenetics, where you're trying to stimulate a particular circuit to modify the activity of that circuit. TMS works in much the same way, and it's much more effective in one of these subtypes than in the others.
And do you think if you looked for those patterns in mice, you could find out for Tony if mice are depressed?
Yeah, we're working on that. One thing I think that's really nice about the mice is that you can, Elizabeth talked about this. One of the limits of fMRI is it's fundamentally kind of correlational in nature. You observe this change in the brain scan, and it seems to correlate with a particular behavior or symptom, but there are many changes in these brain scans, and it's really hard to pinpoint which one, if any, is causally related to particular behaviors, and mice afford us that opportunity. So that's one thing we're trying to do in the brains of our mice. So,
because we want to leave a little bit of time for questions, but so kind of for one final thought for anyone here. So looking down the road, 5-10 years down the road, what's your wish list, or what's your vision of where you think we're going to be, or what do you think is going to be the next, what are we going to do to unlock this and take it the next step, and what are we going to be talking about a decade from now?
Well, I can tell you what we're trying to do. I laid it out. It's the Rosetta brain. I think being able to understand behavior all the way through from the behavior to the neural activity to the actual wiring diagram, for me, is sort of the holy grail to reduce. It's not even reduce, it's to provide the foundation. I went into neuroscience because I wanted to understand how 3 pounds of stuff gives everything that we see, hear, and feel. And it just seemed incredibly mysterious to me. And after being a neuroscientist for several decades, it remains just as mysterious. I don't think we're any closer to...
A little bit?
No, sadly. I mean, the core question, why are we conscious? How does it feel? How is it that we feel the way we do? I don't think we're any closer to understanding. And I sat there thinking about it for a long time and decided that thinking about it wasn't going to get us anywhere further. But I'm hoping that if we actually see an example where we can go all the way from the neural activity through the behavior to the circuitry, it'll at least provide us a way of maybe formulating the right next question, which I currently don't have. I mean, my PhD advisor was Christoph Koch, who has spent most of his career trying to understand consciousness. And I think, and now he's the scientific director at the Allen Institute, where their goals are very similar to the goals of the people here, which is to tease apart the circuitry of a mouse. And so my feeling is that although the circuitry of a mouse is clearly different from the circuitry of a human, basically the building blocks are already there by the time you get to the cortex. If you look at a piece of mouse cortex and you change the scale bar and you put it next to a piece of human cortex, they are indistinguishable. So the basic building blocks, we think, even you're smiling because you're a neurosurgeon, but you will not be able to tell the difference if I change the scale bar. Even the neuroanatomists at the Allen Institute were fooled by this pair of pictures. So I think that we as humans evolved in the last couple 100,000, maybe million years, as primates over the last couple million. I mean, most of the work was done, I think, by the time we evolved as mammals. We split off from mice 70, 80 million years ago. The basic building blocks, I think, are there for us to understand. And once we get those, then I think we're off to the races to figure out what makes us human. But I would be pretty happy if we could get an understanding of what makes us mammal.
So we want to leave a little bit of time of questions. We've got 2 microphones going around, so if you have a question, please wait till somebody brings you a microphone.
Thank you very much to the panel. You've given a really great overview of some of the genetic and biochemical techniques that have advanced the field in the last decade or so. But can you talk more to some of the informatic techniques that might be advancing the field? You mentioned NMF for dimensionality reduction. There have been issues with, fMRI specifically with sort of the kernels and statistics that are going on there. But how has, big data, data science, machine learning also influenced the field? Thank you.
Well, I'll just say that I started off in machine learning and neural networks as a grad student. And I think that that field has affected it in 2 ways. First of all, it inspires us on how to think about how the brain works. Trying to build something is one of the best ways of figuring out how something works. Beyond that, in the last couple years, machine learning techniques have transformed big data in neuroscience just like they're transforming it in many other fields. So that our ability, we're now collecting huge data sets in either calcium imaging or connectivity, and you can't just stare at it anymore. It's the same question of machine learning approaches provide ways of generating hypotheses again, just like knowing the connectivity, and then it takes a real brain. At least so far, we're not quite out of business yet, but these big data techniques are in the process of transforming how we sort of generate the scientific hypotheses that then we can go out and test. That, at least for me, that's one of the big ways.
I would say, coming back again to the other point, is we need computer scientists and people who understand code and big data to understand the brain, right? Because it is a computer, right? And again, the insights you get from people who know that are really critical. What's really fascinating, we've got Amazon and Google and all of those big companies now really interested in brain research. Yeah. They're hiring our graduate students, right? This deep learning, these algorithms, the whole thing is coming together. So I think both fields are now really stimulating each other, and I think that's going to be a path towards getting where we need to go.
Part of the difficulty is to understand actually the behaviors. And the behaviors, it turned out the machine can do a better job than humans in order to determine how many forms of behaviors there are. Actually, Bob Data in Harvard, they are creating those syllables of the behaviors and they're trying to find the neural correlates to that, which it turned out, you actually finding exact the neural correlates to the syllables of the behaviors, which you, as a human observer, you could have completely missing it. So I think that's kind of how these 2 things are, neuroscience and machine learning aspects are really assisting each other.
Another question?
Depressed lab animals produced, developed, isolated. Do you alter the genetics?
How do you produce a depressed animal?
Yeah, I can take that one. The real challenge you mentioned, like animal models of mouse models of schizophrenia, autism. We have a much better handle on the genetics of some psychiatric disorders like schizophrenia and autism than we do for depression. What we know about depression is it's heritable. If your mom is depressed, you're much more likely to grow up to be depressed yourself, and that's not just about environments. But it's probably the result of the shared impact of many, many genes, each having a very small influence on your likelihood of becoming depression, and that interacting with your experience. So that makes it a lot harder to have like a genetically based model of depression that they just don't exist yet. They may one day.
I'll push back on that a little. There's a model of one model of depression is a model of called learned helplessness. And there was a guy who bred generation after generation rats that showed this trait of learned helplessness. And over the last, I think it took him a good 15, 20 years to breed these. Unfortunately, he started doing this when rats seemed like they were going to be the right model system, not mice. But he bred these rats that were genetically distinct and showed this trait of learned helplessness. And he and others have gone on to dissect some of the circuitry that is disrupted in these and identified the lateral habenula, particularly important circuit in this. Now, that's one particular type of depression, but I think it just shows how the kinds of behaviors that we have can actually often be modeled in a rodent. I'll also add to that to say that you can even study things like confidence in a rat or a mouse. It's not obvious that something like confidence, which feels like it's an internal state, would have a neural correlate. But one of my colleagues at Cold Spring Harbor has identified a brain region and particular types of neurons that seem to encode an animal's confidence. And confidence is, for example, one of the things that can be easily disrupted in depression. So I think that when you actually really think hard about how to probe the behavior in a way that traditionally people who work on rats and mice have not, but are now starting to, you can actually sort of get more traction on these things than you might have thought. The sort of ridiculous notion of a depressed mouse or rat becomes less ridiculous. So,
just another question.
Thank you for the presentation. You talked a little bit about how these advances impact the diagnosis of mental disorders. How do you see, on the practical level, what do you see these advances, such as optogenetics or anything else that you talked about here, how do you see them advancing the treatment? Like, what can we expect in the next couple of years with things like ECT or kind of anything that I might not even be able to understand from your answer to this? Thank you.
I think getting back to the transcranial magnetic stimulation thing as one example of a treatment that's based on stimulating a brain region, ECT, electroconvulsive therapy, is our most effective by far treatment for depression, and it's basically a big jolt that is to the brain that's very stimulating to everywhere in the brain, right? And yet it also has a lot of undesirable side effects, and so we reserve it for the people who most need it, who don't respond well to other treatments. I think one potentially exciting avenue in future would be figuring out whether there are particular, more focal brain areas that require a particular kind of stimulation. You could even imagine maybe a little further down the road, cell type specific stimulation, getting at what a lot of Tony's work was describing. The overall goal being more precision in the way we're stimulating brain regions might lead to more effective antidepressant responses and fewer side effects. And I think that's a vision that everyone kind of shares.
So unfortunately, we are running out of time. We really need to thank these amazing scientists for being here.